Hi. Hi, hello, my dear friends. Welcome to the world of zoology. Myself, Dr. Sai from Zoology Wala. My dear future medicos, today don't miss this session because I am going to explain you about the mechanism of muscle contraction. My dear friends, in order to understand this video, you have to follow the two previous videos. You have to listen the structure of muscle, then only you will have an idea about this mechanism of muscle contraction. So this is a very good diagram. I think you might have seen this diagram in your CBSE textbooks. Okay, and now let us see the mechanism of muscle contraction in brief. Okay, so what is mechanism of muscle contraction? See, muscle contraction can be explained with the help of a theory called sliding filament theory. What is that theory? Let me write with this color. So mechanism of muscle contraction can be explained with SLIDING, sliding filament theory. What do you mean by this filament? I explained in my previous class that this is the thin filament, and these are nothing but the Z lines like this. These are the Z lines, isn't it? And here in the center you can see the thick filaments like this. So these are the thick filaments, which is the thick filament is made with a number of myosin molecules. Nearly 200 to 300 myosins forms one thick filament. I already explained in my previous video. So this is the Z line, and this one is also Z line, and the place where the thick filaments are present, this is A band and this is I band. This is I band and this is A band. And in the center of A band, H zone is also present. And now filament means these are the filaments, thin filaments and thick filaments. How these thin filaments and thick filaments will move, that is the point. Whether both will move or only one filament will move, whether whether the A band and I band decreases in size or increases in size, these are the aspects we are going to see. And now, my dear friends, see here, whenever the signal given by the nerve cell, who is going to give the signal? The nerve cell will give the signal, see this one, and now the nerve cell, imagine this is axon and these are the terminals of axon. When the nerve cell will release a signal to the muscle, the receptors which are present in the muscle will receive that signal, and if you observe the layer, see this is the layer, this is the layer, sarcolemma is the layer, something like plasma lemma. The sarcolemma shows inward folding like this. The sarcolemma shows inward folding like this, and these foldings are called T-tubule. T for transverse tubule, and on either side of T-tubule, L-tubules, long tubules are present. This is an area where sarcoplasmic reticulum, sarcoplasmic cisternae are present. Cisternae are the storehouses of calcium ions. Here calcium ions are present. So whenever the muscle receives the signal, whenever the nerve cell will give the signal, that signal was first taken by the sarcolemma. Sarcolemma will take that signal and will give to the T-tubule. In turn, the T-tubule will give signal to the please don't mind. So the sarcolemma will receive the signal, will give to the T-tubule. In turn, the signals will be given to the cisternae, which are present on either side of T-tubule. So cisternae, T-tubule, cisternae forms the triad system, TRIAD, triad system. Portal triad is different, triad system is different, and this is triad system. Okay, T-tubule will give signal to the cisternae. Cisternae releases the calcium ions. If you remember, I explained in my previous session regarding the thin filament. If you observe the structure of thin filaments, there basically, if you observe, say these are all the globular proteins combined to form filamentous protein, isn't it? So these are all the thin filaments, and the thin filament contains active sites. I will show you the active sites with this color. See, imagine this is an active site. These are all the active sites which are present over the thin filaments, but these active sites are not going to be unmasked. when these active sites will expose, when the active sites expose, then only the thick filament will go and bind. When the active sites will expose, that is the point. I explained that in the last class. Here there will be another protein called tropomyosin is present like this. This is tropomyosin, and the tropomyosin contains 3 subunits. What are these? These are the subunits of troponin. Troponin is present like this. So now you have to remember very carefully, active sites are the sites which are present over the thin filaments. They are not visible. Why? Because they are covered by, masked by another protein. What is that? This hand is a tropomyosin, and the tropomyosin contains troponin at regular intervals. So troponin and the tropomyosin are going to block the active sites. See, this is thin filament, and here intermittently active sites are present, and now these active sites are going to be covered by the tropomyosin with the troponin. So tropomyosin with troponin are going to block the active sites. That's the reason why active sites are not completely exposed all the time. When the signal given by the nerve cell reaches here, then only active sites will expose. If active sites exposed, then only the thick filament. See, this is the head. Already I explained in the last class that this is the head of myromycin. This is also the head, isn't it? This is the myromycin molecule I explained in the last class. So the head of myosin will go and bind with the thin filament when the active sites exposed. Then only the cross bridge will form, then only the thin filament can be pulled, or the thin filament movement is observed. It means whenever the muscle wants to move, whenever we have given the signal to the muscle to move, see here now, I am giving the command to this hand, come on, fold, it is folding. Come on, unfold, then it will unfold. So whenever the signal given, then only the muscle undergo contraction and relaxation like this. So here the muscle is moving because of the signal. Unless and until the signal given, the muscle should not move, isn't it? Or else we will be like this only in the restricted form. Our muscles will be like this always, contracted, severe contracted state like this. Okay, when there is no signal, then what happens? The muscles will not move. When we will give the signal, then the active sites which are present over the thin filaments will be exposed. When active sites will expose, then only the head of myosin will go and bind. When the active sites will expose, as I said, when the signal given by the nerve cell taken by sarcolemma, sarcolemma in turn gives signals to the T tubule and then to the cisternae. Cisternae releases the calcium ions into the sarcoplasm. I already told you in the last class, among these 3 subunits, one subunit, this is TPC, meant for calcium ions. When calcium ions reaches to this TPC site, I told you in the last class or in previous sessions, TPC is the guest house for calcium ions. Guest house means only calcium ions live temporarily there. When the calcium ions reaches, see here the calcium ions are present. When calcium ions released into the sarcoplasm, they reaches to this thin filament, and this is an area where the cisternae are present, and here calcium ions are released, they reaches to the thin filament into the TNC or TPC, and when calcium ions comes to these active sites, when calcium ions comes to this TPC, and now what happens, active sites will be exposed. So when calcium ions reaches to the TPC, and now the tropomyosin and troponin complex which is hiding, which is covering the active site, and now it will be unmasked. Who is going to unmask the active sites? Tropomyosin along with the subunit of troponin is going to unmask the active sites. Then only active sites are visible. Why the active sites are not visible previously? Why? Because calcium ions are not there, and tropomyosin and troponin are hiding the active sites. That's the reason why you can't see the marker here. Why? Because these 2 hands are present. What are these 2 hands? Tropomyosin and subunit of troponin are going to mask the active site, are going to mask the marker. If you want to see the marker, the 2 hands should be displaced. If you want to see the active sites, troponin and tropomyosin should be unmasked. So the troponin and tropomyosin should displace, then only the active sites will be exposed. When active sites exposes, then the head of myosin will go and bind. Hope so the point is clear. So it is an interlinking story, very simple. In the examination point of view, sequence model question is possible. How the signal will come to the muscle, I already explained. You can just rewind and you can see all the bits will come in the sequence format. Number 2, who is going to mask the active sites? Tropomyosin and subunit of troponin. When calcium ion reaches there, then troponin and subunit of troponin and tropomyosin unmask the active sites, and now the active sites are exposed. When active sites exposes, now the head of myosin will go and bind with the the exposed active site. See this diagram. For binding it requires energy. Who is going to give the energy? I already told you in previous class, the head of myosin contains ATP binding site. When the ATP will comes here, hydrolysis of ATP takes place, results in releasing of ADP and inorganic phosphate. Along with that, energy also will be released. So hydrolysis of ATP causes the energy, and with the help of energy, and now the head of myosin will become ready to bind with the active site. See here now, the head of myosin already bond with the thin filament. See what is the difference between, imagine this as the first diagram, and this is the second one, third one, fourth one. So if you observe the difference between first diagram and third diagram, first diagram is like this, difference, there is no binding of the thin filament and thick filament. Third diagram, thick and thin filaments bind, forming the cross bridge. And now if we observe the fourth diagram, what happens? See here now, whenever we turn on the switch, the fan will rotate, the fan will move. Why? Because the signal passes like this. Signal always passes in unidirectional way. Similarly, active sites exposed here, active sites, and now the signal is passing like this. So active sites exposed here, here, like this. Active sites exposes continuously. And now the head of myosin, now because of the exposure of active sites, head of myosin bind here, and now the active sites exposed at another site. In order to attach to this new site, the head of myosin should detach from the old site. So it is, see here now, it is not detaching like this. This is attaching. This is not detached. See this is not detaching. Okay sir, this is detaching, but this is not how the head of myosin will detach. Say this is the head of myosin, and now the head of myosin will rotate like this, and now the new active site is exposed. It detaches from the previous site and now attaches to the new site. Why? Because active site is exposed here. So the head of myosin will go and bind with the thin filament, and now the head of myosin will rotate like this. See the rotation of head of myosin. So previously the head of myosin is like this in the third diagram, and now in the fourth diagram the head of myosin became like this. Say this one. This is rotation. So now the thin filaments are present there. Because of this rotation, thin filaments were being pulled towards the H zone. So now see this is the head of myosin. Head of myosin will bind. So many heads are present. I told you minimum 200 to 300 heads are present in one thick filament. Some heads are directed like this, some heads are directed like this, some heads are directed like this, some heads are directed like this. So all the heads will move in this direction. So all the thin filaments will move towards the H zone. All the thin filaments will move towards the H zone like this, So previously the thin filaments, and now imagine my phase is nothing but the H zone, and now these two are thin filaments, and now because of the pulling of thin filaments, what happens? See here now. So this is the H zone. My facial zone is H zone, and now see what happens. This is thin filament. I can show in any direction. Say this one, and now the visibility of my face reduced, and now decreased. That's it. H zone disappears also when the muscle undergoes the severe contraction. So in severely contracted muscle, say whenever we are moving the hand like this, biceps, the muscle is moving like this. So whenever the head of myosin is rotating and bringing the thin filaments were being pulled over the thick filaments. Thin filaments were being slided over the thick filaments. So if you observe, this is the thin filament, and now see the thin filament now here. Previously it is like here, but now it will move towards the H zone. That's the reason why, see the thin filaments are moving like this. Obviously thin filaments are attached to Z line. So Z lines also will come closer, causing the reduction in the size of sarcomere. Let me show you with one diagram. I will take one eraser. Now see this one. Why it is called sliding filament theory, I will explain. It's a very nice image. I will draw one very nice image. See here now. See this, imagine these are thin filaments. Like this. Okay, and here you can see the H zone. I am not going to change the color, it is time taking, okay. And now see what happens now. So this is this line, this is A band, and now in the next diagram what happens when the muscle undergoes contraction. See what happened. This is the contracted muscle, this is Z line, and these are nothing but the thick filaments. So these are thick filaments. And now what happened? See the difference between the first diagram and second diagram what I am representing now. So this one thick filament, this one thick filament. See the previously where the Z lines are present, of course this is I band and this is A band and this is I band. Previously you see the length of A band is constant, the length of A band is constant, but see the I band, say this one, say this one, say this one. Previously observe the width of I band and now the width of I band decreased, and the center A band, the thick filaments are maintained to be constant, but the thin filaments were being pulled. See the thin filaments were being pulled, That is the reason why the I band decreased in size, the length of A band remains constant. Why the length of A band remain constant? Because imagine this is tail and this is neck and that is head, like this. See the myosin is not moving like this. This is not the movement. Myosin is simply moving like this, the head of myosin, okay, like this, like this, like this. For example, imagine you are taking water from the well with the help of the rope. Say this one, binding, pulling, and then detaching. Binding, pulling, detaching. So thin filament binds with the active sites, pull it, and then it will detaches and binds with the another active site. That is the reason why this is called binding, pulling, detaching, or is also called walk along mechanism, something like walking of human being, okay. After attaching the leg to the ground, we will pull the body like this, pull the body, and then the detachment of leg from the ground, like that, walk along mechanism. Say this one, like this, like this. Number of thick filaments are present and number of myromycins are present, 300 myromycins are present. So all the heads are moving like this, number of signals, as a result of which all the thin filaments are being pulled like this. See this, after this rotation, this is also called power stroke with the help of energy. Why? Because it requires it requires energy. So thin filaments were being pulled. Previously thin filament is present here and now the thin filament comes to towards the H zone. After the power stroke is over and now ADP and inorganic phosphate will be released at a time as per CBSE textbook. ADP and inorganic phosphate movement you can see here, and now there is no energy with the head of myosin. Now the pulling is over and now the head of myosin binds with the thin filament very firmly in the absence of ATP. When a new ATP molecule will come, then the detachment will take place and again attachment will take place. So one ATP molecule will be utilized for breaking with the old active site and for binding with the new active site. This is the point you have to remember. So many questions are possible. What are the reactions which takes place before hydrolysis, after hydrolysis, and where the cross bridge formation? What are the events which takes place before cross bridge formation, after cross bridge formation? And ADP and inorganic phosphate will released after cross bridge formation or before cross bridge formation, like that. So many statement model questions are also possible. Generally ADP and inorganic phosphate will be released after the cross bridge formation and after even the complete power stroke is over. A new ATP molecule causes the dissociation. Hydrolysis of ATP causes the production of ADP and inorganic phosphate. Along with that, energy is also produced with the help of which it is able to go and bind with the active sites, and this is power stroke. Pulling of thin filaments towards H zone is nothing but the power stroke. So that is the reason why the length of I band decreases in size. If you observe the severely contracted muscle, what happens? See this diagram, and these are the thick filaments. These are the thick filaments. The length of thick filaments constant, but what happens to the width of the I band? That is the matter. That is matter. See now, like this. See here, and in this image, what you can understand what this image means? See here, these are the thick filaments. In the center, thick filaments are present. These are the thick filaments, and see here. See the length of I band. See the length of I band in the width of I band in case of a first diagram Observe the second diagram, observe the third diagram. And here the H zone, and here H zone decreased, and H zone even disappear also in severely contracted muscle. So which of the following events takes place during muscle contraction? Means disappearance of H zone also observed in severely contracted muscle. The width of I band decreases, the length of sarcomere decreases, Z lines come closer, and the width of A band remains constant. That's it. So one ATP molecule causes the binding, forming cross bridge, movement, and then detachment. Like that, all the thin filaments will slide. The length of the thin filament also remains constant. It is just sliding. It is not contraction. Actually, muscle never undergo contraction. Just it shows the effect of contraction. That's it. So thin filaments will slide over the thick filaments. That is the reason why this is called sliding filament theory, which is proposed by Hugh Huxley and Gene Hansen. Of course, 3 more great scientists are also there. Thin filaments slides over the thick filaments. That's the reason why this is called sliding filament. It's a theory, it is not law. So many objections are also there regarding the sliding filament theory. Anyhow, that I will explain in the coming sessions. That's all about the muscle contraction. Hope you enjoyed, and how the signal will be given by the nerve cell. That is very important. Who is going to receive the signal? Sarcolemma. And once everything process has been over, now the muscle undergo relaxation. In order to relaxation, all the events should become reverse. Calcium ions again goes back to the cisternae. Again active sites will be masked, and again once the active sites will mask, now the head of myosin will detaches. Whatever the events I explained will become reverse in the relaxation of the muscle. That's all about the muscle contraction. We'll see in the next video. Thank you, my dear friends. All the best.